So how can we blast more than just the genome sequence at SoyBase? Pictured here is the SoyBase home page, and we have a section for a quick blast of the Williams-82 genome, which allows you to just paste in a single sequence and do a blast against only the pseudo-molecules. Well, if we go up here and look at this link that says full blast and click it, you'll be taken to our advanced blast page. You'll be given a number of options. The first of which is a choice of how you want to see your blast report, either in the context of all 20 chromosomes or in the usual blast report. You also will be given an opportunity to pick the appropriate program for your input sequences that you're going to use as a query. And then you'll be able to pick a number of databases that we have available. We have the Williams-82 genomic sequence, plus we have the coding sequences and transcript sequences, as well as protein sequences from the 2 genome annotations. We also give you a list of a number of soybean sequences that you can blast against that came from GenBank, and we also have the organellar sequences, the chloroplast genome, and the mitochondrial genome, along with their individual gene calls and protein sequences that came from GenBank. We also have the chloroplast and mitochondrial scaffolds that came from the Williams-82 sequencing project. We have the database of soybean transposable elements developed by Ma et al. 2009. And we also have a database that contains all of the genes that have been identified so far as being deleted in the fast neutron mutants described by Bolin et al. In 2010. Blasting against this with your gene sequences will allow you to determine whether they've been knocked out in a mutant, and you might be interested in that mutant. So down a little further, we have the option to change your filtering. Then we also have a number of preset expectation values that may be conveniently spaced, but if those aren't sufficient, you can come down to our advanced options part of the page, where you can choose a number of options, including the ability to type in -e space 1 e to the negative whatever number for your expectation value that you want. And finally, you are able to pick from a number of alignment views that you're probably now used to from blast reports that you see at GenBank, and you have the ability to limit the number of descriptions and alignments that you get back. And at this point, we also want to point out that we give you the ability to upload from your computer a multiple FASTA file containing a whole bunch of sequences which will be blasted one after the other in one convenient blast report. This has been a SoyBase production. For more information about SoyBase, go to www.soybase.org.